Okay, so now going to chapter 7, going to talk about linkage, recombination, and eukaryotic gene mapping. I am not going to go into a lot of details in chapter 7. I think it's way too deep and too complex and too mind-bugging, and is more of a graduate level type of chapter. Okay, so we're just going to look at the very basics and what exactly you need to know out of this chapter. Go ahead and read the chapter if you want to. I read it and I got confused. So if you focus in this, you know, it's easy, targeted, exactly to the point of what you need to know. First idea is that a lot of times genes don't come alone. They come with some other genes that they like to hang around with. So this is a story of linked genes. You know, usually people who have red hair will also have freckles. And it's very rare. Actually, I've never seen a person that has red hair and no freckles. So these genes are very close together in the chromosome. When one goes, the other follows. Okay? So these alleles here, for example, red hair is big A and freckles big B. They'll always stay together. Either you have it or you don't. You're not going to have big A, little a. The chances of that happening are very, very low. So those are the linked genes. They come together as a unit. In this case, you do not have independent assortment. So first you have to start thinking, how is it that the genes, the alleles, are going to get mixed up in the first place? We know here that the only way that you can mix up the alleles is if you have crossover in what stage of meiosis? Prophase 1.
C is for clue, that's okay with me.
It showed up before, it's going to show up again. So you get crossover in prophase 1. Very important to know that. That's when you can cross over the genes from one chromosome to another. Right, you get mixing up. Now, if these genes are very close together, chances are that you are not going to get crossover. So if you look at these genes here, you have, you know, A and B that is far down on the chromosome. One is very close to the centromere and the other one is further down the road. When you have the crossover here, if they're, you know, far apart, chances are they're going to get mixed up. But when they're very close together, okay, here if you see A and B and C here, most likely A, B and C, this one and these ones here are going to get crossover, so you can have this type of combination. Now B and C here are very close together, so when you get the crossover, they're going to shift as a unit. They're just going to stick together. The closer they are, the more stuck together they're going to behave like. So how do you figure out how close the genes are to each other? You can do this genetic mapping by number of recombinant frequencies. If you see that two particular genes are recombining all the time, so you can say that they are far apart. If they are never recombining, you can say they are very close together, okay, like the red hair and the freckles. So far apart, recombine all the time, never recombining, very close together, and then you have everything in between. Sometimes combines, sometimes doesn't combine. The frequency of combining, if it is very high, so chances are they're very far, and then it's very low frequency of recombining, chances are they're very close. So you just look at the number and solve the puzzle. First thing you have to do is figure out which genes are further apart. Now, when you look at the recombinant frequency, you have to look which one is recombining the most. Okay, here B and C is recombining the most, 36% of the time, let's see. So you can put B and C in the two streams on this chromosome. Right? So the next farthest one is what? C and D. The number is pretty high, 23, right? So now you have to figure out, where am I going to put the D here? I put B and C, they're far apart. C and D, okay, is 23. It could be C is bigger separation. You look at, let's say, which one is the next one here? D and C, and then D and B. Oh, D and B here, you look, is 13. So D is closer to B than it is to C. Right? So you just put in the map somewhere that's going to be closer to this side and not to the other side. And just like, you know, solving the puzzle, you just put them in there in a way they will make sense. Next one, A and B. Now, where is A and where is B? They're not there. So let's say A and B is 21, is very far apart too. So you put A farther from B, about 21, is all in proportion, right? The next one here is going to be what? A and C is 15. So you see, you have to look at your A and D. A and D here is 8, they're close together. A and C 15, so okay, put the A somewhere in between there. It still makes sense. Now B and D 13, yes, it still makes sense. And the last one is going to be A and D, which is the smallest separation, is 8. So in this case here, just based on how far they are from each other, you can figure out their position on this particular chromosome. Is that simple? Yeah? Piece of cake? Okay. So we're done with chapter 7.